And now we have Dr. Douglas Wallace, somewhere, who will share with us some of his fascinating work in the field of human mitochondrial DNA genetics. In addition, he has made seminal contributions to human and mammalian genetics, and his work has contributed immensely to the understanding of human migration and human evolutionary history. So it's with great pleasure I introduce Dr. Wallace. Thank you.
Well, I want to thank the organizers for inviting me and for all of you to be here. And I guess this is laser. Yeah, so I'm going to start from a different task, which is why can't we understand and cure the common metabolic and degenerative diseases? Those are enormous number. Neurodegenerative, neuropsychiatric disorders, the heart and muscle diseases, visceral, metabolic diseases, cancer, aging. And it's not because we haven't spent hundreds of millions, billions, trillions of dollars on trying to understand these diseases. It must be that there's something else wrong. And what might be wrong? Well, we have to start with a concept that Thomas Kuhn put forward, which is when scientific development begins to stagger, then maybe the problem is not the effort, but in fact the basic assumptions on which the scientific community is addressing the problem. So what are the basic assumptions that Western medicine is using to look at the problems of metabolic and degenerative diseases? Well, the first assumption goes back to Vesalius, and that is that diseases are organ-based. So if I have a headache, I'm referred to the neurologist because the assumption is if there's a symptom in my head, there's something wrong with the head. Okay? I call that the anatomical paradigm of disease. And the other basic assumption goes back to Mendel, and that is that if something is inherited according to the, quote, laws of Mendel, it's genetic, and if it's not, then it must be environmental. And these ideas then are the framework on which all medical students are taught and all basic scientists try and design experiments. But that may not be sufficient. It is true that all the Mendelian genes code for all the anatomical genes, so that was very simple. We could understand then the specific disease mutations in specific genes which gave rise to the newborn screening program. But it doesn't seem to be helping us with these common complex diseases. And so what's the problem? Well, the problem is that it's not just enough to have an anatomy. One has to be animated. And you're the most animated thing in my environment. And so there was a fellow named Newton who said that mass doesn't change unless it's acted on by energy. So therefore, if we're going to think about being alive, we have to think not only about anatomy, but also energy. And we need to think about the information for anatomy and information for energy. So if we then begin to think this way, then in fact maybe half of medicine is about energetics, not just about anatomy. And so that leads us to the hypothesis that bioenergetic dysfunction may lie at the nexus of the genetic environmental causes of the common complex diseases. So how does this dichotomy of anatomy and energy come about? Well, it actually came about from the origin of the very cells that make up your body, the eukaryotic cell. And that was a symbiosis between an archaeobacteria and an oxidative bacterium, so-called an alpha proteobacteria. And the archaeobacteria ultimately gave rise to the nuclear cytosol, and the oxidative bacteria gave rise to the hundreds of bacteria that live inside each of your cells called the mitochondria. And these two bacteria have their own information storage and retrieval systems. The nucleus has DNA that's transcribed into RNA. The RNA is translated on cytosolic ribosomes, giving 20 to 25,000 proteins, about 1 to 2,000 of which go to make the anatomy of the mitochondria. But the mitochondria has retained its own DNA, and that DNA is transcribed replicated and transcribed in the mitochondria, and the messenger RNA is translated on mitochondrial ribosomes. And those ribosomes are bacteria-like, so translation is initiated by N-formyl methionine, just like bacterial proteins are. And these ribosomes are sensitive to chloramphenicol and aminoglycosides, just like bacterial ribosomes are. Well, what happened is that over time, these two originally co-equal genomes began to specialize. And the nuclear genome specialized in creating the anatomy of the cell, not only the anatomy of the cell, but also the anatomy of the mitochondria. And the mitochondrial genome specialized in energy. And it developed the wiring diagram for the power plant. So just like power plants around Washington, D.C., how to build the power plants with the city manager, but all the wiring diagrams of each different power plants is different. And so therefore, those wiring diagrams are in each of the specific power plants. And that's exactly how your body is designed as well. So the mitochondrial DNA codes for critical proteins that are involved in the energy process called oxidative phosphorylation. So it codes for 7 of the 45 proteins of what we call respiratory complex 1, 1 of the 11 proteins of complex 3, 3 of the 13 proteins of complex 4, and 2 of the 15 proteins of complex 5. So what are, in fact, these complexes? Well, these complexes are a system by which the cell takes the nutrients in your diet and the oxygen that you're breathing and converts it into potential energy that you then use for everything that you want to do. And so looking just at glucose, as we heard before, glucose goes through glycolysis to give pyruvate. Pyruvate can be reduced to give you lactate, or it can get amino group to give you alanine. But pyruvate goes through the pyruvate transporter, and pyruvate dehydrogenase to make acetyl-CoA and drive the Tca cycle. Now, the purpose of the Tca cycle is to strip the hydrogens off the hydrocarbons and put them on the carrier NAD, and NADH then has the 2 protons and 2 electrons of 2 hydrogens. And these hydrogens are burned by what's called the electron transport chain, complex 1, CoQ, complex 3, cytochrome C, complex 4, and the electrons flow down from reduced to oxidized, where they then react with oxygen to give water. Now, the energy that's released is not just dissipated, it's used to create a capacitor. So as electrons flow through complex 1, 3, and 4, they pump protons from inside the mitochondrial matrix across the mitochondrial inner membrane to the intermembrane space to be positive and acid on the outside and alkaline and negative on the inside. So sitting in your chair right now are 100 trillion cells. Each of those cells has 1,000 bacteria, and those 1,000 mitochondrial bacteria, 10 to the 17th bacteria in your cell, and that's the, about 30% of your body weight. Now, each of those mitochondria has a potential across the membrane of about 0.2 volts. So the total energetics in your body right now is the equivalent of a lightning bolt. And that is the energy for everything that you do every day of your life. And if you don't think this is important, stop breathing for the next 15 minutes. Okay? So this flow of energy is absolutely critical. Without this flow of energy, you become a cadaver, you're inert, and you go to medical school, but in the wrong way. Okay, so you can use this membrane potential for lots of reasons. One thing you could do is you could regulate a positive cation. So calcium is taken up by the mitochondria because it's electroporesed into the mitochondrial matrix, which is negatively charged. But it can also be used to make ATP, and that's through complex 5, the ATP synthase. So the protons flow through a proton channel in the ATP synthase to condense ADP and phosphate to make ATP. And the ADP and ATP are exchanged across the inner membrane by the adenine nucleotide translocators, which then go out and are phosphorylated to do work. So you're coupling oxidation with phosphorylation. We call that oxidative phosphorylation. Now, the mitochondria also has, is like any furnace, it makes smoke, but the smoke are reactive oxygen species. That is, not fully oxidized electrons go to O2 to give you superoxide anion, hydrogen peroxide, and this can be reduced to water by nicotinamide nucleoside transhydrogenase that makes NADPH, and glutathione peroxidase that reduces hydrogen peroxide to water. If you don't, in fact, get rid of the hydrogen peroxide, then transition metals will give you another electron to give you hydroxyl radical, and these are the reactive oxygen species and the potent damaging agents, and why some of you take vitamin C, vitamin E, and beta-carotene, or CoQ. The mitochondria also has a self-destruct system, which is actively debated what structure is, but its job is normally to be closed door, and then when the membrane potential gets low, or high energy phosphates decline, or oxidative stress gets excessive, or excessive calcium load occurs, then it impinges on this system which senses those things and ultimately pops into an open channel which short circuits the membrane potential. Fluids flow in, the inner membrane swells, backs and back form a megachannel, out go the pro-apoptotic proteins, and they degrade the cell from inside out. Why bother? Because these are bacteria. If they're released into the bloodstream, they have all the bacterial antigens and they will create inflammation. And that's why we believe all the metabolic and degenerative diseases also go along with inflammation, because without enough energy, apoptosis fails. So the mitochondria then generates most of the energy. It regulates the redox state of the cell. It makes the reactive oxygen species, which are signaling molecules, but at high levels are toxic. It regulates calcium. It regulates apoptosis. It generates all the intermediates for regulating the epigenome, ATP, acetyl-CoA, acetylmethionine, alpha-ketoglutarate, et cetera, and different tissues in your body have different energetic demands. So the brain is about 3% of your body weight, uses 20% of all your mitochondrial energy. So therefore, a 10% reduction in mitochondrial energy is going to give you a very bad headache. It's not because your brain has altered, it's because there's a systemic defect and your brain is specifically affected. So the hierarchy of energetics are brain, heart, muscle, renal, endocrine, and liver. And those are the organs that are affected in all the common complex diseases. So why would the mitochondria then retain these 13 proteins? Because there's 7, 1, 3, and 2, because those are the electron and proton wires of the wiring diagram. And all of these proteins have to co-evolve, because if any one of them became leaky for protons, it would short the capacitor. So therefore, all of the enzymes of this system relying on the same substrate, that is the membrane potential, have to co-evolve. So that all the wiring diagram is an integrated circuit. Now, how would you do that, since nuclear genes are all undergoing recombination? So if there were polymorphisms that affect complex 1 but not 3 and 4, then you could have recombination that would mix and match, coupled and uncoupled variation. And what do I mean by coupling? Well, if you're very efficient at taking calories, burning them and making a membrane potential, and then converting that membrane potential to ATP, then you will burn the least number of calories for the maximum amount of ATP. And a calorie is a unit of heat, so you'll generate the least amount of heat. But if you're less efficient at pumping protons out or converting them to ATP, then you're going to have to burn more calories for the same amount of ATP, and that's going to generate more heat. So your mitochondria is regulating your thermal and your energy balance based on the coupling efficiency. So then you can see that having a loosely coupled mitochondrial system means you're going to eat more calories, and therefore you're going to remain thinner on a high-calorie diet. Okay. So anyway, why would then the mitochondrial DNA retain these? Because, of course, as they are the wiring diagrams. But as I was saying, if these were all nuclear genes, then they would be mixed and matched, and the coupling efficiencies would be all scrambled up. So Mother Nature was very concerned about that, and she thought a lot about it, and she said, well, the problem is men. So, in fact, she decided to put the wiring diagram in a DNA that's inherited only from women. And then there could never be a mixing between men and women. There would never be recombination, and women would have all the power, which we already know is true. So, the mitochondrial DNA is transmitted from mother to all of her children, and her daughters to their children, whereas males' mitochondria enter the egg, are seen as foreign, and are selectively destroyed. So, guys, don't feel bad. Women have thrown you out for 2 billion years. Okay. So the mitochondria has thousands of copies of the mitochondrial DNA, the cell does, and the mitochondria are constantly replicating inside your cell and being eaten up by mitophagy. So it's a colony in steady state. And as they replicate, mutations accumulate, and those mutations then create a mixed population of mutant and normal mitochondria. We call that heteroplasmy. Now, if the cell divided down the middle, then both cells would get some mutant and some normal, and that they would both be heteroplasmic. But if the cell divided this way, then this cell would have only normal, and this would have twice as many mutant. So mitochondrial genotypes segregate during mitosis, and therefore the tissues in our bodies are a mosaic of different mitochondrial genotypes, with different tissues having different percentages of mutant and normal mitochondrial DNAs. As the number of mutant mitochondrial DNAs increases, the energy output declines until it crosses the minimum energy for that organ, and now you begin to have a metropolitan brownout, and you have the equivalent of an energetic disease. And we believe all those diseases that I was talking about are bioenergetic diseases. Mitochondria has a very high mutation rate, 1 or 2 orders of magnitude higher than nuclear DNA, so there's a tremendous amount of genetic variation. It falls into 3 categories. 1, mutations that arise along the maternal lineage. These are maternally inherited diseases, and these are just a few examples. Here's a mutation in the ND4 gene that if you inherit that, this is a nucleotide position 11778. If you inherit that from your mother, you'll be fine throughout until midlife, and suddenly you go blind in one eye and then in the other, called Leber's hereditary optic neuropathy. Mutation in the ND6 gene at 14484 or at 14459. 14484 gives you that same Leber's blindness. 14459 is more severe. When it's heteroplasmic, it gives you blindness. When it's homoplasmic, pure mutant, gives you generalized dystonia. Mutation in the ATP-A6 gene at 8993 at 70% mutant gives you retinitis pigmentosa, 85% olivopontocerebellar atrophy, 90% kills you as an infant with Lee syndrome. So we have a quantitative genetics that gives you quantitative energetic deficiencies. You can also have protein synthesis mutations. Oops, sorry. Mutation in the tRNA leucine gene at 3243. At 20% mutant will give you diabetes. At 50% gives you cardiovascular disease. At 100% mutant is lethal in childhood. Mutation in the tRNA glutamine gene that accounts for about 3 to 5% of late onset Alzheimer's and Parkinson's disease. Mutation in the tRNA lysine gene will create a kind of epilepsy, myoclonic epilepsy. And then there are all kinds of cancer mutations, which we won't go into today. So the second class of mutations are ancient polymorphisms. So this variant in ND1 gene that is found in 3/4 of sub-Saharan Africans, and we call that macrohaplogroup L. Variant H is found in half of Europeans. Variant A, B, C, and D arose in Central Asia. They crossed the Bering Land Bridge and allowed people to colonize the Americas. And we believe these are markers for mitochondrial lineages that adapted human energy metabolism to live in different environments. And finally, as we age, we're accumulating somatic mutations, and that is the aging clock. Okay, so once we begin to think energetically, then all the common diseases have the same etiology, a bioenergetic defect due to oxidative phosphorylation. You can have nuclear mutations that affect this process or changes in the epigenome. You can have ancient adaptive polymorphisms or recent deleterious mutations that affect OXPHOS. The calories that you eat and the type of nutrients you have, or how you exercise and how you use those nutrients, or whether you smoke and the toxins, they all impinge on energetics. If you affect energetics, then you affect mitochondrial DNA replication, and you accumulate mitochondrial DNA damage over time, and that gives you an age-related decline, and that's what aging and delayed onset and progressive course of all the common diseases is due to. If you have a partial defect in energetics, it's going to affect the brain, the heart, the muscle, and the renal system, and those are all the diseases we talked about to begin with. And if you impair the furnace, you're going to build up substrates, which is glucose, fatty acids, cholesterol, and that's metabolic syndrome. And finally, when mitochondrial energetics is damaged, then in fact apoptosis fails and the bacteria are released in the bloodstream, and that initiates all of the inflammatory systems that goes along with complex diseases. And finally, cancer is all about adjusting energy based on nutrients and oxygen. So in fact, a bioenergetic way of looking at the disease takes us away from the anatomical approach and into an energetic approach to medicine. So this is a family we worked on in the early 80s. This woman had lactic acidosis and growth retardation. All of her children that we could examine had lactic acidosis, growth retardation, progressive dementia, stroke-like episodes, hypertrophic cardiomyopathy, cardiac conduction defects. The mitochondrial oxidative fibers degenerated in the muscle, but the glycolytic muscle fibers were fine. These individuals had a mutation in the tRNA leucine gene. They were about 70% heteroplasmy, and they had myopathy, cardiomyopathy, and stroke-like episodes. But this exact same mutation, when present in 10 to 30% mutant, can either give you autism or type 1 or type 2 diabetes. So those are classified differently, but in fact they can be caused by exactly the same mitochondrial point mutation. And if you have 100% mutant, you'll die as an infant with Lee syndrome. So we wanted to know how a change in mitochondrial heteroplasmy in a cell could affect these different phenotypes. So we made what are called cytoplasmic hybrids, or cybrids. These are cells with the same nucleus but with different percentages of mutant mitochondrial DNAs. We then took those cells and characterized them physiologically, which I won't go into, but then we did RNA-seq and looked at all the transcription factors that were regulated in these different cell lines. And so 20 to 30%, which is diabetes, these are the transcription factors that are on-off. 50 to 90%, which is neuromuscular disease, that's the array. And 100%, which is the lethal childhood array, that's it. And then a cell with no mitochondrial DNA That's the pattern. Or put in principal component analysis, this is normal, these are diabetes and autism, these are neuromuscular disease, these are lethal childhood disease, and that's a cell with no mitochondrial DNA at all. So what does that say? It says that the phenotype is determined by the mitochondrial signaling to the nucleus, and the signaling to the nucleus determines gene expression, and the gene expression determines the phenotype. So in fact, the mitochondria is determining what the environmental challenges are and telling the nucleus how to respond, and that's telling the physician what he or she will see in the clinic. So this is a pedigree which has a mutation in the ND6 gene, and I'm going to quickly go back because I forgot to tell you what the genes were. The mitochondrial DNA codes for a small and a large ribosomal RNA and 22 transfer RNAs that punctuate the genes. ND1, 2, 3, 4, L4, 5, and 6 are complex 1 genes. Cytochrome B is complex 3 gene. CO1, 2, and 3 are complex 4 genes. ATPase 6 and 8 are the 2 complex 5 genes. Okay. So now back to this particular mutation. This is in the ND6 gene, or complex 1. This woman had 50% mutant in her white blood cells. She had optic atrophy, cerebellar ataxia. Her sister had 5% mutant in white cells, and she was perfectly normal. She had 3 consorts. Every one of her children was 100% mutant, and they all died. This shows the rapidity of replicative segregation that occurs along the germline, and why this class of disease was never understood by a Mendelian conceptual framework. So this particular mutation then changes proline at codon 25 to a leucine. And so we wanted to prove that this was pathogenic. So we took cultured mouse cells, we mutagenized the mitochondrial DNA, and then sequenced all the mutants that were respiratory deficient and found an array of different mitochondrial DNA mutations. So one of them has exactly the same mouse mutation that we saw in that human phenotype. Another one is cytochrome oxidase mutation that changes valine, codon 421 to alanine. Or if we just mix 2 perfectly normal mitochondrial DNAs, what do we get? So how do we do this? We take the cell with the mutant mitochondrial DNAs, we remove its nucleus, we take the cytoplasmic fragment with the mutations. We made a female embryonic stem cell, we kill its mitochondria with this drug, and then substitute the mitochondria with the mutant mitochondria by cell fusion. We then have these cytoplasmic hybrids, which are pluripotent. We put them into blastocysts, put them into foster mother. Now we get chimeric females and breed the female for transmission of the dominant agglutinin locus through the oocytes and pick up the mutant mitochondrial DNAs. Okay, so this is the mouse with the proline 25 to leucine in ND6. This is the normal optic nerve. This is the mutant optic nerve. You see increased axonal swelling. You see demyelination because these patients also have multiple sclerosis. You get abnormal mitochondria. And what was interesting, if we look biochemically at the synaptosomes of the brain, the problem is not in the ATP. The ATP was the same. There was about a 30% reduction in respiration, but if you increase the energy demand, respiration will go up to near normal. But what happens is not that it affects the ATP, but the reactive oxygen species production goes through the roof. So this is a disease of oxidative stress, and that kills the neurons. This is the mouse with the mutation in cytochrome oxidase. It has a 50% reduction in cytochrome oxidase in all of its tissues. It gets the ragged red fibers that we saw in that original family with abnormal mitochondria. It gets progressive cardiomyopathy with fibrosis, and as it ages, it gets type 2 diabetes. So we've again recapitulated these same phenotypes with single point mutations. But this is the most interesting. We just took 2 perfectly normal mitochondrial DNAs, mixed them together, and with the same nucleus then segregated the heteroplasmic animals back out to the homoplasmic 129, NZB, and the heteroblasmic form. And then we asked, did it matter whether they were heteroplasmic or not? So we looked at activity. Mice are active at night, not during the day, and the 129 mitochondrial DNA animals are active at night, but not in the day, and the NZB are active at night, not in the day, but the heteroplasmic animals just sit there. They're depressed. And if we ask those animals to learn a task, here we have an open field with different holes around the outside, and just one of them has a little black box, and they have different colors here so that mouse can learn. So you put the mouse in the open field, and it doesn't like being out and exposed, so it looks around at all the holes till it finds the box and it jumps in. And it then learns, based on the color cues, how to find the box. So the 129 and the NZB animals learn over successive days, and so does a heteroplasmic animal, but it learns slower. But then you wait 2 days after the 4th trial and put the animal back. The 129 animal immediately runs and jumps in the hole. NZB animal jumps in the hole. The heteroplasmic animal has learned nothing. It has no long-term memory. So simply converting a homoplasmic cell to a heteroplasmic cell of perfectly normal mitochondrial DNAs has severely altered the neuropsychiatric pattern of those animals. So what are these mitochondrial lineages? Why is 129 and NZB mitochondrial DNAs different, and how does that relate to you? Well, in fact, these are about as different as an Eskimo would be from a Kalahari Desert bush person's mitochondrial DNA. So where did these mitochondrial DNA variations come? Well, in humans, mitochondrial DNAs arose about 200,000 years ago in sub-Saharan Africa, and the original mitochondrial DNA lineage is L0. From there, L1 and L2 arose, and then from L2, L3 arose. Now, how do I mean by this? Well, mitochondrial DNA can only be transmitted from mother to daughter. So the only way it can change is by sequential mutations. So if I sequence the mitochondrial DNA of any two of you, the number of changes is equivalent to the time you shared a common mother. So by sequencing the mitochondrial DNAs from people around the world, we can then reconstruct their genetic relationship and therefore their migration pattern. So again, the Khoisan people have the most common ancient mitochondrial DNA lineage. These are two pygmy lineages. These are sub-Saharan Africans. And then in Ethiopia, we discovered that two lineages arose, M and N, and only these two mitochondrial DNAs left Africa and colonized the rest of the world. M went into the temperate zone and gave rise to a whole bunch of European lineages, which we gave letters I, J, T, U, U, K, I, W, X. And then also went into the temperate zone in Asia, whereas M stayed in the tropics all the way to Australia, and then very much later acquired new mutations to live in the temperate zone of Asia, and then lineages C, D from M and A from N became rich in Chukotka, and they, 20,000 years ago, crossed the Bering Land Bridge and colonized the Americas. So why is that astonishing? Because nuclear polymorphisms are found panmictically throughout all populations. But mitochondrial DNA variation is highly geographically constrained based on the geographic origin of your ancestors. And that's why 23andMe can do your mitochondrial DNA and call you Ursula and say you came from Sweden. Why? Because we showed that, in fact, that U lineage arose in Sweden. Now, why would it be that there would be this amazing geographic contribution to regulating mitochondrial variation? Well, because here in Africa, if you're a woman and you want to reproduce, you need to run away from lions. Running away from lions means that you need to have a lot of ATP. Therefore, all of your calories need to be tightly coupled to maximize your ATP. And it's hot, so you want to minimize the heat. So that's fine. But up here, the lions froze to death. That's not a problem. The problem is you're going to freeze to death. So how did you not freeze to death? You have accumulated mutations that decreased your efficiency of your mitochondria. Now you're eating more calories for the same amount of ATP. What does that do? That increases your thermal regulation, your more heat. So that's why these people at a high-fat diet kill marine mammals, and that's why we in Washington look down on them. But I'm sorry, that's their niche. Anyway, the point is that all of these that are adaptive mutations that allow our ancestors to live in different environments and cope with different things, infection, diet, environment, thermal regulation, etc. So what are some examples? So this is just a very tiny part of the European lineage. Every time there's a branch in the mitochondrial DNA tree, it's founded by a highly functionally significant change in a protein. So this lineage J is founded by two cytochrome B mutations, 15257, 14798. If we look at these mutations, 14798 is conserved in all Mesozoic animals, but it's polymorphic in this population. 15257 is conserved all the way to E. coli, but 5% of the people in this room have that polymorphism. That's unheard of. That's antithetical to what we think about evolutionary biology. If something's conserved across evolution, it should be homogeneous within a population. Not so for mitochondrial variation. Mitochondrial variation is variable within species, even though it's homogeneous across species, because it's our adaptive engine. It allows us to adapt our energy to environmental changes. Okay, so this is a mutation that arose 10,000 years ago in Europe in the tRNA glutamine gene, an A to G mutation. It's found in only 0.4% of modern Europeans, but it represents 3% of AD, 5% of PD, and 7% of AD-PD patients. There are also single mutations that arise that are much more deleterious, and this is the mutation ND4, methionine 31 valine, that gives you AD-PD immediately. Okay, this is a study just looking at European autism patients. And what we find is that, in fact, mitochondrial variation correlates dramatically with autism by these mitochondrial haplogroups. And this counts for 55% of the European population's risk of autism. So mitochondrial variation accounts for a whole bunch of diseases: neurological, metabolic, inflammatory, altitude adaptation, cancer, aging, athletic performance, etc. Now, this is a nuclear mutation that affects the adenine nucleotide translocator isoform 1 that's found in heart, muscle, and brain. And this mutation arose in this pedigree. This is a Mennonite pedigree, and the mutation arose in Switzerland about 500 years ago. These populations came to North America carrying this recessive mutation, and then eventually heterozygous individuals married each other, and they give rise to affected homozygous mutants, because this is a frameshift mutation in the ANT1 gene on chromosome 4. But what's interesting is that these people have very mild cardiomyopathy, whereas these people die of fulminated dilated cardiomyopathy. And what's the difference between them? This shows then the heartbeat of cultured cardiomyocytes derived from embryonic stem cells, and this shows the heartbeat of these stem cells in a normal individual, and if we have the mutant individual, they're highly dysrhythmic. So once we know then that this is affecting cardiac dysrhythmia, then why is it that we have this defect or differential in phenotype? The differential is because those people with the severe cardiomyopathy have European lineage U, and those people with mild cardiomyopathy have European mitochondrial lineage H. So it's the mitochondrial DNA that's determining the severity of the disease, not the nuclear mutation. So we can then make a mouse that has a mutation in the ANT gene, and these animals then start, wild type will exercise. This is showing oxygen consumption, and till they stop running, till exercise gives up. But the mutant animals start running and fall down, and they have a progressive accumulation of abnormal mitochondria. They also have the highly dysrhythmic cardiomyopathy as opposed to the wild type. And now what we can do is mix the CO1 and the ND6 mutations with the ANT. If we mix ANT and CO1, we don't get a big significant effect. But if we mix ANT with the ND1, we get very, very severe cardiomyopathy with half the lifespan. So again, you can see how the mitochondrial DNA is determining the phenotype, not the nuclear DNA. So now we can ask, what do these mouse mutants tell us about behavior? So we take these mice and we simply ask them how they respond if we put them in a constraint for 30 minutes, a very mild response. And so the mitochondrial DNA mutations, the ND6 responds much more strongly with corticosterone response than the CO1 or the wild type. And if we look at the nuclear mutation, the ANT has a huge response, whereas the antioxidant enzyme NNT is severely limited in this response. So here is the ANT on looking at catecholamines, and you can see whether stressed or not, it has no effect. This animal is already freaked out, like me. Okay, so now we can ask, does this affect neurodevelopment? So I'm almost done. And that is, we have two types of neurons, an excitatory glutamatergic neuron and an inhibitory GABAergic neuron. And the glutamatergic neurons are born at the base of the brain and migrate radially. They don't go very far. But the inner neurons, the GABAergic neurons, are born at the base of the brain and migrate tangentially. So now we can ask, in the developing mouse, what happens when we have a mutation in the ANT? Well, the wild type, all the neurons are migrating fine, but the mutant are lost. And we can use an enzyme inhibitor, binucleic acid. Without the inhibitor, they're going tangentially. With the inhibitor, they're going radially. So what this does is it makes the animals hypercompulsive, very bad nest builders. And this same thing is seen for the ND6 mutation and for the heteroplasmic animal. So these mitochondrial variants are also affecting brain development. So the last point to be made then is we think the mitochondria are the environmental sensors. When the environment changes, they change the epigenome by all these mitochondrial intermediates, and that reconfigures the mitochondrial genotype to maintain homeostasis. But if the environmental challenge is too great or there are mutations in the nuclear cytoplasmic system, then they can't adjust, and that gives you energetic deficiency and disease. And so what we're doing in China is we're taking advantage of the fact that for 5,000 years, China's medicine has been based on the idea of qi, which is basically vital force. And we are anatomical in our view, but the mitochondria forms the bridge between anatomy and energetics. And so we're now looking at all the traditional Chinese therapeutics from a mitochondrial point of view to look at new therapeutic approaches. And these are all the great people that this has worked. Thanks.